Welcome everyone to lecture 13 of BIS103. Today we'll shift gears a little bit. We want to actually pause for a moment on learning about new pathways, right? We already have discussed a number of pathways. We have seen how many of these pathways can flux in different directions, such as glycolysis versus gluconeogenesis. We have seen how all of these pathways, and or at least many of them, cooperate with each other and intersect at certain reaction steps. So it's a good time to just pause for one lecture and start looking at how we actually achieve control over all these different pathway directions and functions, right? How can we regulate all these different pathways, the cooperations between them, and especially how can we have control over the direction of the pathways, for example, whether we want to use glycolysis or gluconeogenesis. So we want to look specifically at enzyme regulation and cycling of pathways through regulation. So we have a number of learning goals today that we have to work through. First and foremost, we want to be able to explain why controlling, regulating flux through metabolic pathways is important. Then we want to be able to describe two important concepts in regulating metabolism: futile cycling and reciprocal regulation, and why they are important to metabolism. We want to explain and we want to be able to provide examples for two kinds of enzyme regulation: allostery and covalent modifications. And then we also want to be able to apply that knowledge of pathway regulation to understand which are actually the key enzyme steps, the key reactions in these pathways where we want to regulate these pathways. And last but not least, we want to be able to describe the role of key hormones and that they play in metabolic regulation. So we have a bit of a task ahead, but let's dive into it. Let's start first with why. Why do we want to regulate metabolic flux? What's the relevance here? And there are two major reasons you could think of. One is you only want to form products when you need them, right? You only want to make glucose when you're in need of glucose and not all the time. So being able to regulate those pathways is really helpful. The other one is to control the use of pathways that are fluxing in opposing directions. Again, we can use our example of glycolysis and gluconeogenesis. Right? You want to make sure that you don't run them at the same time, because then you would be breaking down glucose at the same time as you're making it. So in general, you want to prevent that. We'll actually see examples of why we're in fact doing that and what the advantage of that is. And so in general, and there are exceptions, but in general, opposing pathways such as glycolysis and GNG do not occur at the same rate at the same time. That's what we call complete futile or complete substrate cycling. Generally, we want to prevent that because what you're doing, right, is you have net product outcome, right? If you're fluxing them at the same rate, meaning same flux and the same time, you're breaking down the product just as fast as you're making it. All you're doing is that you're using up ATP and potentially your electron carriers and cofactors. So you don't seemingly achieve anything here, right? So in general, they don't occur. Complete futile cycling usually does not happen. Okay. If you remember, we actually had one example of futile cycling when we talked about our TCA cycle, and specifically about the fact that animals, unlike plants and microbes, cannot bring about the net synthesis of glucose from fatty acids as a carbon source. Right? And the reason was, like, if you remember our TCA cycle here, that we brought in acetyl-CoA as a 2-carbon unit, and we had these two decarboxylation reactions. So we have a plus minus 2, we have no net carbon gain that we could use for making glucose. Right? Now, to run this as a theoretical example, we had said, right, if we wanted to make glucose through GNG, we would likely use OAA as a substrate using our PepsiK enzyme that we had seen before. So this seemed fine. But we had also said, right, if you take out a metabolite of the TCA, you have to refill it. So our anapleretic reactions had to kick in, and we only have one available here to bring OAA back. And so in this case, you would need pyruvate for the pyruvate carboxylase to produce OAA and refill the TCA cycle. But my pyruvate, the way we get pyruvate is PAP through our pyruvate kinase, the last step of glycolysis. But then we need PAP that we also here need for GNG, right? So this would run as a futile cycle, essentially, right? You wouldn't achieve any product in a net manner. All you would do is use up ATP. So in general, we want to avoid doing this because we're not really gaining anything. So generally, you can assume that you want to prevent complete futile cycling. And because of that, enzymes that are catalyzing reactions of the opposing irreversible steps into opposing pathways are regulated, and they're regulated in these opposite directions. So they're regulated reciprocally. If you again take the example of glycolysis and gluconeogenesis, right? If you have the irreversible reactions and you have one forward reaction, one reverse, you would activate the forward, for example, you would reduce the reverse one in the same way. So you would reciprocally regulate both those enzyme steps running in the opposing directions. Now, what's interesting is that while complete futile cycling really does not help, with very, very few exceptions, partial futile cycling actually occurs very commonly, and I can assure you, you're doing it right now watching this video. And so partial futile cycling actually means that the opposing pathways occur at the same time, but, and this is really important, not at the same rate. Right? So you would flux the forward reaction, for example, more than the reverse reaction. So they're happening at the same time, but not at the same rate. And yes, it's true, that does waste, if you will, some ATP, but we will see in an example that I'll show you in just a second that what it does is it allows for greater and for dynamic control over the pathway flux in both directions. And that can be really helpful for metabolism. So why is it advantage? Why is partial futile cycling advantage? The example I want to use here is actually glycolysis and gluconeogenesis, and specifically our two irreversible reactions in the opposing directions, our PFK1 here, right, our irreversible reaction 3 of glycolysis, converting fructose 6-phosphate using ATP to fructose 1,6-bisphosphate. And then in gluconeogenesis, right, we had seen the way we overcome this irreversible glycolytic reaction is using our fructose 1,6-bisphosphate phosphatase here, that is dephosphorylating fructose 1,6-bisphosphate to fructose 6-phosphate. So two enzymes in the opposing directions, both irreversible reactions. Right? Let's now think about some scenarios between complete and partial futile cycling to see how this might be advantageous. Okay. So what I illustrate here now is essentially the rate of flux. So I'm just using some numbers. They're not coming from any experiment. They're just arbitrary numbers to highlight just the different rates of flux in this case. And what I want to show is the flux in direction of PFK1, so glycolysis, and the reverse reaction of the phosphatase in gluconeogenesis. And then here just the net flux that we're achieving under these different scenarios. So our first scenario would be no futile cycling, right? So let's assume we are running PFK1 in with a flux rate of 10. Right? So say per minute, we are generating 10 molecules of fructose 1,6-bisphosphate. So that's the rate of 10. We don't do any futile cycling, so our reverse pathway, GNG, is not running at all. So it's 0, meaning our net flux is 10. Okay. So now let's look at complete futile cycling. So we're running both pathways at the same time, same rate. So again, our forward rate for PFK1 is 10. We're doing complete futile cycling, same rate, so 10 in the reverse reaction, and your net is 0, right? So this, flux-wise, seemingly really does not have any advantage to us, right? But let's look at partial futile cycling, right? Say I'm running the PFK at a flux of 100, okay? But I'm doing partial futile cycling, so same time, but not the same rate. So say I'm running the reverse reaction, the phosphatase, at 90. Okay. What that gives you now is a net of 10 again, which is exactly the same as if you were doing no futile cycling. Okay. Still doesn't seem to make a lot of sense, I would assume at this point, right? All you're doing is you're running different fluxes, but your net is the same, right? But now assume you want to make a really rapid dynamic change in your flux, right? What happens then, right? So assume you see a bear and you want to run away, right? Some of you may remember last year we actually had a bear who got lost on campus. So assume you were running around at rec hall, you saw the bear, you wanted to get out of there. You have a rapid dynamic change in your demand on glycolysis, right? You want to make energy so you can run away. Let's see how this works between different concepts of fuel cycling, okay? So you have paid some attention, BIS103, you understood complete fuel cycling really doesn't help, but you don't want to do partial fuel cycling, so you don't do any fuel cycling. But you want to run away, so you increase your glycolytic flux, so PFK1, and you double it, so you get to 20 here, right? Now you don't do fuel cycling, so your GNG is not running at all, so there's nothing to regulate. It stays at 0, right? If it's not running, you cannot do much with it, right? Your net is 20, so you got a little bit more energy to run away. Now, for those who have really paid attention to BIS103, right, you said, okay, there might be something about partial fuel cycling, so let's try this out. And I do the same rate of increase, I double it, right? But I ran my PFK1 at 100, so I go up all the way to 200 now. I also have been running, right, my reverse reaction here with a rate of 90, and because I've been doing that, I can now reduce it, right? If you don't do it and it's at 0, there's nothing to reduce. But if I've been running it as part of partial fuel cycling, I can reduce it. So say I doubled my forward reaction, so I halve my reverse to 45. You net, right, 155. So I'm making a lot more energy than those who decided not to do fuel cycling. So you can imagine who runs away faster from the bear, right? So this is just an example, really, of how partial fuel cycling, while it is using up some of your ATP resources, can be an extremely helpful tool to bring about very quick and very dynamic changes in metabolic pathways. And because of this, we are actually doing it all the time. Organisms are constantly running some level of partial fuel cycling to maintain this dynamic control. So as a take-home message, right, partial fuel cycling overall provides greater control over pathway flux. All right. So now that we have an understanding of why it's important to regulate pathways, let's have a look at how we can do this, right? How can we regulate metabolic pathways? There are multiple levels of regulation, and we don't have the time to cover them all. Generally speaking, you can say that there's actually regulation happening at the DNA level, there's regulation happening at the mRNA level. Those we won't focus on here. They're covered in your physiology and cell biology and genetics classes. The other level here is the regulation at the protein level, and that's what we want to focus on in this lecture. And so again, there are a number of ways how regulation can occur at the protein level. Mostly we're looking at regulating enzyme activity. Okay? And there are a few ways that we can do this. We can change, for example, the enzyme amount. So we can either make more enzyme. This would intersect to some degree with regulation at the mRNA level, or we can degrade it. So simply changing the number of enzyme molecules that catalyze a reaction that you want to change. We could look at also looking at different types of enzymes, so isozymes that have different catalytic properties. So that would allow us to, for example, change activity. So using enzymes with different catalytic rates, for example. And the third one, and this is actually a really important one, they're all important, but this is a specific one that is used widely in metabolism, is to change enzyme-specific activity. So not changing the amount of enzyme in any way or the type of enzyme, but changing the activity of the one enzyme that is already present. The advantage of this type of regulation is that it's very quick, right? You can imagine that just simply changing the activity of an already present enzyme is much faster than having to either make a whole lot of more new enzyme or breaking it down. So the change here is a way of very effectively, very rapidly changing and regulating metabolic reactions. There are two types of how we can achieve these regulations of enzyme-specific activity. Those you really want to remember. The first one here is allosteric effectors or allostery, and the second one is covalent modifications. What we want to do next is to look at the concepts of these two and provide a few examples. So let's start with allosteric effectors or allostery. What this is that you can change enzyme activity and you can activate it or inhibit it through non-covalent interactions of a metabolite usually, so a small molecule, with the enzyme. Okay. And so there are different names for these. They're called effectors, modulators, or most commonly allosteric regulators. Again, non-covalent interactions and usually involving small molecules, metabolites. What is really critical for the definition of allosteric regulation is that this effector here, so the small molecule, will bind somewhere in the enzyme other than the active site. Okay. So these are not inhibitors that are attacking the active site, they bind somewhere else. If you want to remember it, allo actually is a Greek word for other, so it might be sort of a little brain help here to remember what this means. In addition, the fact that this is non-covalent means also that it is very easily reversible, and because of that is very fast. So these effectors can just reversibly quickly bind to the protein and be released again, and it's concentration dependent, right? The more of these effectors you have, the more of these enzymes can be impacted. Allosteric regulators are very, very common, and they act in probably all pathways. I'm actually not aware of any exceptions. They can act within one pathway at multiple steps, for example, so they're extremely dynamic, extremely widely distributed. One of the key roles of allosteric regulators actually is to maintain ATP and cofactor balance, right? All quarter now we had talked about how important it is to maintain a base level of ATP and to maintain a balance of cofactors throughout all our different metabolic pathways, right? And so these regulators, one of their key functions really is to maintain the ATP and cofactor balance. If you think about this, right? So if your prime directive, as we call it here, is to maintain ATP levels and cofactors, then using small molecules, using metabolites that have something to do with energy levels might be a really smart thing to do. And this is in fact what is happening is that there are few allosteric regulators that we would call energy-rich sensors, so metabolites that tell the organism, I have a lot of energy, and some that are energy-poor sensors that do the opposite. So these are your alarm signals that say, I'm really running out of energy. And so these are really good metabolites if you want to regulate pathways that have to do with ATP and cofactor maintenance. So before we move on, right, if you want to do this at home, you can maybe stop the video just for a second and think about all the pathways we had discussed so far and which of these intermediates and products we had talked about might function as such sensors. So here are some examples, right? As energy-rich sensors are ATP itself, right? If we're making a lot of ATP, we don't really have a need to make more, right? We have a lot of ATP to do biochemical work. That would be an energy-rich sensor. NADH, the same, right? If NADH accumulates, it would mean that you actually don't have much of a requirement to run your ETC. So that would sense, yeah, I seem to have enough energy. Likewise, citrate, right? Our first intermediate of the TCA cycle, right? If citrate accumulates, it would signal that, well, I don't seem to have to run the TCA cycle a lot. Seems I don't actually need a lot of NADH. Seems to me I have enough energy. Right? So these are really common metabolites that are part of energy metabolism and really function as these energy level sensors. And then the opposite is true for some of the metabolites that indicate low energy, right? And these are the opposite ones, right? This would be your ADP or your NAD+. And specifically AMP here, right? I'm had mentioned it several times during the quarter now that AMP is sort of one of the really critical alarm signals for I am running out of energy. And remember, right, your adenylate kinase here that can take ATP and AMP and can reconvert it into 2-ADP in order to maintain the substrates we need for ATP production, right? We have just seen that in our lecture on ATP synthesis. So AMP is actually a key sensor for poor energy, for low energy. And these are really just a few examples and to highlight them for you. And just drive this fact home, right? Here I actually just show sort of the overall pathway coming from glucose down our catabolic, metabolic pathways are here to pyruvate or PDH reaction, and then the TCA cycle here. And you can see right here, we show in green, these are activator steps. In red here, these are inhibitory steps. But you can see that all these energy sensors, in fact, are allosteric regulators for all these different reactions that we already had discussed. It gives you an idea like how dynamic these regulations are and at how many steps we're actually applying these energy sensors as regulators, right? It's extremely complex and extremely dynamic. So we'll only sort of look at a few examples to try and highlight for you how the concepts of these regulatory mechanisms actually work. So let's look at the first concept. How is allosteric regulation actually achieved? All right. So there are two different kinds. It can be inhibition or it can be activation. Right? So if we start right up here, this is an example for an allosteric inhibitor. Right. So you have your enzyme here, right? And down here, this is your catalytic subunit. So your active site, that's where substrate to product conversion would be happening, right? And here's your substrate that could go in. The enzyme does its job. Up here now you have a second domain, so a second part of the protein with another binding site, but it's far away from the active site, right? Remember our definition of allostery, that it has to be a binding spot other than the active site. So now imagine, right, your allosteric compound comes in, your small molecule, it's inhibitor, so it will bind at this position, and this binding will change the conformation, so the shape of the active site, and now the substrate will actually not be able to enter the active site anymore, no catalysis is happening. So this is one example on how small molecules, allosteric regulators, can inhibit an enzyme function very quickly. In the opposite way, you can also activate enzymes in the same idea, right? So assume that in its native form, this same enzyme now here would have a, say, closed active site, so the substrate can't enter, nothing's happening. Now you have, again, an allosteric regulator. In this case, it's an activator. It binds to this enzyme domain here outside of the active site. It does facilitate a conformational change, so a change in the shape of the active site, but in this case, it would open it up. So now your enzyme becomes active. So you're using the same principle, but for different outcomes, whether you want to inhibit it or activate it. And the reality is that many enzymes can actually have one or more activators and inhibitors. So there are examples where you have far more than a dozen different metabolites acting as allosteric activators or inhibitors for one enzyme. And so thinking about inhibition and activation, there are two different kinds, right? Two that we define are the feedback inhibitors and the feed-forward activators. So we want to see some examples of those now and how they work. So let's start with the feedback inhibitors up here. So these are compounds that act as inhibitors to prevent the substrate binding and the catalysis of an enzyme that is upstream in the pathway. So a reaction that has occurred earlier in this same pathway, okay? So let's just use sort of a hypothetical pathway here, right? And so we're running this pathway, and we're making product E, and at some point, we just have enough of it, it starts to accumulate. We do not want to make more of this product. So now we actually want to shut down this entire pathway. So now we're actually using our metabolite E here, and we're using it on our first enzyme in the pathway, right? And so that's just highlighted here. Now you have, right, this is the active enzyme, the substrate is bound to the active site. Now product E comes, it actually binds to the allosteric binding site of this enzyme, and we're changing the conformation of the active site, we're inactivating this enzyme now. And so it's a feedback inhibitor because the product downstream in the pathway is actually inhibiting substrate conversion very early on in the pathway. And this makes a lot of sense, right? Because if you want to shut down this pathway, you want to prevent using any kind of resources that you don't need to use. So, right, if you would only block this enzyme here, which also can happen, but just to assume it here, if you just would do it here, you're still running all the first three reactions, but you don't really need to because you don't want to make this product, right? So it makes sense to go back as far as possible to prevent the use of any metabolic resources if you do not want to catalyze this reaction. So that's what we call feedback inhibition. Okay. So if you want to sort of just have, again, a little brain help here, you can imagine it as, right, say there was an accident here somewhere down the highway and the police is coming in and is actually starting to slow down traffic. Sort of the same idea, you're looking ahead, you're seeing there's an obstacle, you're seeing there's something you don't want to do, and so you come in and you're slowing down everything in the back. Sort of just as a little help, hopefully that will be useful somehow. All right, now to the opposite, right? How about our feed-forward activators, right? How does this work? And one example actually is in our glycolytic pathway that actually fructose 1,6-bisphosphate. So one of the intermediates of our glycolytic pathway is actually a feed-forward activator of the pyruvate kinase, our last step. So it activates and it increases the activity of our last step of glycolysis. Right? So the same idea here, looking at traffic, right? Essentially, if you imagine now you're sitting at a light, it goes green, and the person in the front is on their phone again or sleeping again, what you have to do is, right, you have to honk so that they get going. So that's the same idea here in our pathway, that, right, if this is accumulating, it indicates, well, we have to push through this pathway now, we need to make more, and we need to free up all these other enzymes. So this metabolite now acts as a feed-forward activator to increase the activity of the last enzyme of the pathway, to remove all the obstacles and increase the overall rate of the pathway. Right, so now that we understand these two different concepts of feedback inhibition and feed-forward activation, let's have a look at some examples of these metabolites. And I'm using as an example here our glycogen phosphorylase B. We had seen this in our breakdown of internal glycogen. Right? That was a phosphorylase that we saw there. And so if you actually look at it, right, there are a number of metabolites here that function as either inhibitors or activators in an allosteric fashion. Right? And so here is ATP, for example, that's an inhibitor. Glucose 6-phosphate is an inhibitor. AMP is an activator. Now think back to this concept of energy sensors and think about if this makes sense, right? So let's start with ATP and glucose 6-phosphate. ATP is a high energy sensor, right? If you have ATP accumulating, we obviously don't need to break down more resources to make more ATP. If you have glucose 6-phosphate accumulating, it's in a way the same thing, right? It's an intermediate in your glycolytic pathway. So if it is accumulating, it means you're not actually running the entire pathway through the ETC. So you're not removing glucose 6-phosphate. So both sensors for high energy. If you have high energy, there's no reason to break down glycogen. Right? So it makes sense that these are allosteric inhibitors. On the other hand, right, AMP, our low energy warning signal, is an activator. So if this now tells you I have AMP, excuse me, I'm running out of ATP, so there I have AMP now, My apologies. So this makes sense now, right? You're running out of energy. You want to break down glycogen to free up glucose that you now can run through the glycolytic pathway, PDH, TCA, and the ETC, right? So oftentimes, really, you can already think about energy metabolism, and you can make good suggestions based on that alone on what kind of metabolites might be allosteric regulators of an enzyme. So this covers the allosteric regulations. Now let's look at the other type of regulation, the covalent modifications. And so unlike our allosteric regulators, these are now modifications that really do covalent additions of a functional group in an enzyme. So we're actually making concrete changes to an enzyme. We're not just reversibly attaching a metabolite. The first important aspect to say here is that this requires enzymes to do it. Okay? So if you want to covalently change an enzyme, you need another enzyme to do this. So this will be the first time that we're seeing enzymes that don't have small molecule metabolites as their substrates. Their substrates are other proteins. Okay? And so there's a number of these types of covalent modifications. We only will actually focus on the first one here, which is phosphorylation. Right? There are a number of others, anilylation, urylation, methylation, and so forth. That's not even the entire list here. But we want to look at phosphorylation here. That actually happens to be one of the major ones, if not the predominant one, to modify enzyme functions through covalent modification. And so again, I want to give you an example. And so we're using, again, our phosphorylase B here, okay? And now we're not actually using allosteric regulation. We want to use covalent modifications. And what we're doing is we are phosphorylating our phosphorylase, right? So again, this requires a second enzyme that has to catalyze the phosphorylation of our glycogen phosphorylase. So up here, you have your phosphorylase. And just focus on our hydroxy groups here, right? These are serine side chains. Now your kinase comes in, and yes, it's a kinase, like we had seen in many of our other reactions, but it's not converting a metabolite, it's converting a protein, but it's a kinase because it's catalyzing a phosphorylation using ATP as a donor of the phosphoryl group. So this takes ATP to do it. And we're phosphorylating the hydroxy groups of these serine side groups. Okay. And so what you're seeing now is, right, in this process, in its non-phosphorylated form, this phosphorylase is less active. Once you phosphorylate these amino acid side chains, you are making it more active. So one form of changing the enzyme activity through covalent modification, in this case, phosphorylation. Don't get confused here by the B and A. Initially, when the Corys actually found these enzymes, when they're working on the Corey cycle, they saw these differences in activity and thought that these are actually two different enzymes. And so they called them phosphorylase A and B. Now we actually know they're the exact same enzyme, but in its phosphorylated and non-phosphorylated form. So don't get confused. They are the same enzyme, but they are modified forms thereof. All right, so we'll see this phosphorylase a little bit later, but let's first look at reciprocal regulation. What does this mean, right? We had touched on it earlier already, that we had said opposing pathways are regulated reciprocally. What does this actually mean? Okay. So again, in general, right, we want to prevent complete futile cycling. And because of that, enzyme activities are regulated reciprocally through allostery or covalent modification. So what this means is now, regardless of which type of regulation you're using, is that you will take the enzyme, say in the forward direction, and you will regulate that. And at the same time, you will also regulate the specific activity of the enzyme or enzymes that are catalyzing the reaction in the opposing direction. So you're looking at both directions, and you're regulating both enzymes that catalyze either of these reactions. And in order to simplify it and to make it efficient, you will actually see that in many cases, you're actually using the same mechanism. So you're using the same type of regulation For example, using the same allosteric regulator to activate one direction and to inhibit the other. So again, just to give you an example here, similar to what we've seen before, right? We have in the one direction here our gluconeogenesis, right? Our fructose bisphosphate phosphatase that I had shown you when we talked about partial futile cycling. And we have the reverse reaction here, it's our PFK1 in glycolysis. Right? And now have a look at our regulators here. Again, the first thing you note is, right, these are all these energy sensors here that are coming in, right? But they work in different reactions, right? So we have AMP here, our low energy sensor, for example. It is inhibiting gluconeogenesis. That makes sense, right? We're sensing low energy, so we don't want to make glucose. We want to use it up to make energy. So AMP inhibits the phosphatase in the direction of GNG, and at the same time, it acts as an allosteric activator for your PFK1, so you can boost up glycolysis so you can make energy, right? So it makes sense. The opposite is not true for example, citrate, right? If you have high citrate, it means you have high energy, no need to do glycolysis, so it becomes an inhibitor. On the other hand, right, if citrate is low, it becomes an activator here of gluconeogenesis to deal with moving glucose along, right? So the same thing true here, these energy sensing metabolites now can be regulators in the opposing directions, having different functions, inhibitory versus activating. There's one here that I highlighted in green that I need to mention, and there are a few allosteric regulators that you cannot simply deduce from their role in energy metabolism. There are some special ones. In a way, they exist because they provide pools of regulation that are positioned outside of your mainstream energy metabolism. And one specific one here is fructose 2,6-bisphosphate. Right? So it's very similar to our glycolytic intermediate fructose 1,6-bisphosphate, but it has a phosphate group sitting at position 2. Okay? And so this is actually a really important allosteric regulator in your liver. This is what it looks like and how it's produced, right? We're actually using our glycolytic intermediate fructose 6-phosphate. We're using a kinase, but this is a kinase that now specifically attaches the phosphoryl group at C2 and not at C1 right here. What we're generating now is a molecule that is very, very similar to your glycolytic intermediate, but it is not an intermediate that will be converted as part of glycolysis or gluconeogenesis for that matter. Okay. The enzyme that actually brings about this reaction is called the phosphofructokinase 2. So it's a sibling to our PFK1, but it catalyzes this other direction that phosphorylates at a different position. Okay. It's a bifunctional enzyme. What that actually means is it has two activities. It actually has two domains, one being a kinase domain and one being a phosphatase domain. So you can see this actually here. So I just illustrate what this enzyme actually looks like. So it's an enzyme here where essentially you have two enzymes that got fused to each other. And on the one hand here, you have the kinase domain. On the other hand, you have the phosphatase domain. And we'll actually see later that we can, much like we could regulate two enzymes in a different way, we can actually regulate these domains in different ways too, even though it's one enzyme. Okay. There are many examples of these bifunctional enzymes. This is just one of them. All right. So now that we have an idea of how we can regulate pathways and why we want to do it, let's look at which are actually the enzymes that we want to target here. Right? We already looked at a few of them, right? Your glycogen phosphorylase, your PFK1, your fructose 1,6-bisphosphate phosphatase. So they might give you a hint already of which are the sort of key nodes of regulation, which are the key reaction steps. But let's look at the logic, the chemical logic behind choosing these enzymes. In general, right, the key steps of regulation, the key reactions where regulation is occurring are our irreversible reactions. Right? So for example, reactions 1, 3, and 10 in glycolysis, your kinase reactions. Also, in general, the reversible reactions are not as much regulated. The reality is almost every enzyme reaction in metabolism undergoes some level of regulation, but sort of really broadly speaking, the key reactions are the irreversible ones, so reactions where you have two different enzymes functioning in the opposing pathway directions, whereas the reversible ones are not as much regulated. The reason for this is that if this is actually the same enzyme, you don't have to use another one because it's a reversible reaction. If you start to regulate this reversible enzyme, you're shutting down both pathways at the same time, and that's no good to you, right? Then nothing is happening anymore. So let's again use our example of glycolysis, right? And our three irreversible reactions, 1, 3, and 10, and have a look at the chemical logic of what we would be choosing here in this pathway scenario. So here's just straight up, right? Your glycolysis going from glucose, first reaction to glucose 6-phosphate, then the reversible one, another irreversible one to the fructose 1,6-bisphosphate. Then there are, right, all these reversible reactions all the way to PEP, and then your last reaction, reaction 10, to pyruvate, another kinase. So you could argue, okay, right, we have learned about feedback inhibition, right, and feed-forward activation, thinking about those things, where here would we attack? We could say, well, just downregulating this one here at the end of the pathway might not make sense because we're using up all of this, the other resources here already, right? So we might not want to choose this one. It will be regulated, but it might not be the most important one because it's so late in the pathway. So you could argue, okay, maybe this first one here, right? Because, well, then we're not using up any resources, right? Keep in mind all our BIS103 maps, right? We also had all these branching pathways, right? For example, here to and from glycogen or glucose 6-phosphate use for the PPP, right? Essential pathways. You don't want to block them. So if you actually use this as a major regulatory enzyme, you're before the branch. That means you're not just shutting down glycolysis, you're shutting down everything else because you're not making glucose 6-phosphate anymore. So that might not be a good idea either. What is actually happening in metabolism that you often strike the compromise. Okay? So the actually most important regulatory step in glycolysis is a reaction from fructose 6-phosphate to fructose 1,6-bisphosphate. It's your PFK1, because it's right in the middle between not using up resources and minimizing that as much as possible, but also not preventing the branching pathways from occurring. So this is your, what we call the K-pi, the key physiologically irreversible enzyme, or K-pi. This, while all three irreversible reactions are regulated here, this is the major one because it allows for the most dynamic regulation of glycolysis here. Okay. All right, so I hope this chemical logic makes some sense, and I would encourage you to look at some of our other pathways like the TCA, PPP, GNG, and try to work out for yourself, where would you think might the KP lie for these pathways? And then I want to move on sort of from this and start to look at some examples, right? How can we apply these concepts to understand the broader theme of metabolic regulation? And I want to use glycogen metabolism as an example. In order to do so, we have to first introduce one more new allosteric regulator. And again, this is one that we don't necessarily can deduce from our energy metabolism, and that is cyclic AMP, or CAMP. Some of you may already have heard about it. What it actually is essentially AMP with the monophosphate group, the phosphoryl group, forming a cycle, a ring, with the ribose molecule. I will not ask you exactly how it's made. It's actually made from ATP. So we're using the hydrolysis of the two phosphoanhydric bonds to bring about this cyclization reaction for a CAMP. And the enzyme that is catalyzing it is the androlate cyclase. So this is, you just want to remember the name and maybe that it's coming from ATP. You don't have to remember the structure. what I want you to focus on is the fact that it's an important allosteric regulator, and we'll see its function in the next slides. And what it regulates is an enzyme we haven't seen yet. That's the protein kinase A, or pKa. Okay. You can already see from the name protein kinase, it seems, right, the function will be that this is a kinase that is using proteins as a substrate. So let's have a look at this. And this should look familiar, right? We just touched on our phosphorylase already when we talked about the covalent modifications of enzymes to change their activity, in particular using phosphorylation. Right? And we had said that the activation of phosphorylase B in glycogen metabolism is brought about by phosphorylation here to activate the enzyme. And we had said we require a second enzyme to catalyze this phosphorylation. And this actually is our pKa, or the protein kinase A. So this is actually the enzyme that is catalyzing this phosphorylation reaction. So keep this in mind, this is its function, right? It's activating phosphorylase A in this particular case, but it's much more dynamic and versatile than only this one reaction, and we'll see this. So let's look at a number of scenarios, and I realize this is a confusing slide, but we'll walk through it step by step. Under a certain scenario, what all is happening and how can we use these different regulatory mechanisms to control our metabolism. And so what I want us to look at is glycogen metabolism, so glycogen breakdown and synthesis, in a scenario where we might want to produce more glucose. So say you've been watching this video, you're getting really hungry, you want more glucose, right? You're running out of glucose, you want to do more. So what is happening here, right? The first thing that we're seeing is that CAMP, or cyclic AMP, is being increased, and we'll see later who is increasing this. And cyclic AMP, right, is our allosteric regulator. It's an activator of pKa, right? So this is allosteric regulation. And now this is activating pKa, and we want to see what's happening. And we want to look at our glycogen breakdown rate down here, and we want to look at glycogen synthesis here. So let's do this step by step. Step one, right? So in the liver and in the muscle, now you're wanting to make more glucose, your cyclic AMP is increased, right? We want to activate glycogen breakdown, right? Glycogen breakdown to generate more glucose. Cyclic AMP now activates our pKa, our protein kinase. Okay. What it does now is that it actually is phosphorylating, right? That's its function. It's phosphorylating our phosphorylase B kinase. So not the phosphorylase yet that we just spoke about, okay? It does that too, but what it actually does initially is that we increasing the activity of another kinase through pKa, making it more active through this phosphorylation. And now this phosphorylase kinase, in turn, it's also a kinase, it's using a protein as a substrate. This now will increase the activity of our phosphorylase, making it more active, again, through phosphorylation. And as a result, we're increasing the rate of glycogen breakdown because we're making the phosphorylase that is breaking off the glucose 1-phosphate units from our glycogen more active. So through this process, we're increasing glycogen breakdown now. Why are we using now like two different kinases? Why not go through this entire process just with one kinase? Right? And the reason here is, why the cascade? The reason is amplification, right? So some of you who may have played Domini before or seen this, right? It's sort of the same principle with just increasing the number of stones that are being moved, you can have a complete cascade effect. And kinases do the same, right? You could imagine that, right? pKa will increase the activity of a number of these phosphorylase B kinases. And each of those activated kinases can again increase the activity of a whole number of phosphorylases, and so forth and so forth. So you have this avalanche effect, which means that the activation process can happen much, much faster than if you were only using one single kinase. So this is actually a very common system that we're using for signal transduction. Some of you may have heard about MAP kinases in signaling pathway, and so it's working exactly the same way there. All right, so now we have used our pKa to increase glycogen breakdown. What's happening to glycogen synthesis now? So it turns out that pKa is actually a multi-substrate kinase, and it can actually convert a whole number of different proteins. And another one that it is modifying is our glycogen synthase. So the enzyme that's actually making glycogen from glucose units. Okay. And so again, cyclic AMP is high. It activates our protein kinase A, and it phosphorylates them. But unlike what we've seen before with our phosphorylase B kinase, phosphorylation is actually decreasing activity. Okay. So what is happening in this case here is we're now slowing the rate of glycogen synthesis. Right? So it's a nice example of using the same mechanism, allosteric regulation by increasing cyclic AMP, and covalent modification by phosphorylation, but we're using the same mechanisms in a different way. We're using these mechanisms for glycogen breakdown by increasing the kinase and the phosphorylase activity, and the same mechanism, aka phosphorylation, for a glycogen synthase. So the enzyme that is catalyzing the opposing pathway is being reduced. So same mechanism, and we're simultaneously regulating both opposing pathways, right? And so we want to make more glucose, so we increase glycogen breakdown here. And obviously we don't want to make glycogen at the same time. That would be futile cycling, right? So we are down regulating this. Both reactions happen in the liver and the muscle. The muscle now has another trick up his sleeve. The muscle actually does an additional thing. And so keep in mind, right, we want to make more glucose here for our entire system. So in the liver only, and the muscle prevents that. It has this trick that it can prevent this. We're actually here now increasing cyclic AMP again. And now we are using our other allosteric regulator here, this fructose 2,6-bisphosphate, and this bifunctional enzyme, this kinase and phosphatase enzyme. Okay. So what's happening now, again, we're activating pKa. It will phosphorylate this bifunctional enzyme. And what's happening is that the kinase activity is being reduced and the phosphatase domain activity is being increased. Okay. So again, right, it's a two-domain enzyme. It has a kinase and a phosphatase domain. Through this phosphorylation, we can interchange the activities of both domains. What is being increased here is a phosphatase domain. If you do this right, if you increase the phosphatase domain, you are decreasing the abundance of our allosteric regulator, right? So we're decreasing the amount. If you now look at it, right, fructose 2,6-bisphosphate was an inhibitor of gluconeogenesis and was an activator of glycolysis. So by reducing the concentration of the allosteric regulator, we are increasing gluconeogenesis and we are decreasing glycolysis. Why this makes sense is, right, we want to make glucose. So in addition to the glycogen breakdown, we're activating gluconeogenesis in the liver to release even more glucose for our system, and we're decreasing glycolysis. There will be some happening for the energy metabolism of the liver alone just to do this, right? But we are favoring gluconeogenesis to bring about the production of even more glucose. Now it might make sense why the muscle has the trick of not doing this, because, right, the muscle does not release free glucose, right? The purpose of the muscle is movement. It wants to do glycolysis. It will not release free glucose, so it will not actually have this reaction occurring. It occurs only in the liver. All right, so now that we have sort of seen one big example on how we can use both allosteric regulation and covalent modifications to really broadly regulate, for example, glycogen metabolism and its catabolism and anabolism, we want to really understand, well, who's controlling cyclic AMP, right? If cyclic AMP is controlling all of these reactions upstream by controlling the activity of our pKa, who's doing the controlling of cyclic AMP? How is it coming that cyclic AMP is being increased? And this is where your hormones kick in. Right? And so the major hormones that you have, you probably have heard of those, right? Is your adrenaline. That's actually a small molecule coming from tyrosine primarily. You have your glucagon here. I always remember it as glucose gone. It's a sensor for low glucose, right? That's a small peptide. And then your insulin, your high glucose sensor, essentially as a hormone, is a little bit larger. It's like a dipeptide here that is combined by these disulfide bonds. Again, you don't have to remember the structure even generally of these hormones. What I want to focus on is their function, and really only in general terms. But what the hormones do is that they now actually regulate carbohydrate, sugar metabolism at the global level by controlling, among others, but specifically the abundance of cyclic AMP. All right, let's look at how this works. Let's start with glucagon and adrenaline. It turns actually out that these in many ways do the same thing, but for different purposes. Right? Adrenaline is your fight or flight hormone. Glucagon is your glucose gone hormone. It's indicating low energy. Both would want to have more glucose released, more glycolysis to be done. You need energy, right? Whether you're running out of energy and glucagon comes in as a sensor hormone, or whether you're in a flight or fight situation, you want more energy, so you want to have glucose to generate energy. So you want high glycogen breakdown, high glycolysis, high TCA cycle, and so forth. And so what these hormones now do is that they bind to receptors on the cell surface, right? So I'm just highlighting here, there are 3 cells, a liver cell, there's a muscle cell here, and an adipose cell, right, for your fatty acid metabolism. We'll focus on them separately. But what is going on here, right, you have these different receptors for glucagon and adrenaline right here. I don't ask you to know exactly how the signaling mechanism happens. We'll actually not talk about it. What I want you to see is that the hormones binding to these cells will trigger a signaling cascade. That cascade will increase the abundance and activity of the adenylate cyclase that produces cyclic AMP. So you're increasing the abundance of cyclic AMP. That now can trigger your entire pKa cascade. Okay. So this happens in the liver. That's one. If we now look at our receptors in the muscle cell, right, we do have our adrenaline receptors. That makes a lot of sense, right? In a flight or fight situation, you want your muscle to be able to do glycolysis and generate biochemical energy so you can run away or do whatever you have to do. But you might see that makes sense that it doesn't have glucagon receptors. Again, right, the muscle is not useful for us to generate free glucose. It is not releasing free glucose. That's the job of the liver. So the glucagon receptor, which is our low glucose, low energy receptor, and is triggering release of free glucose to your system. The muscle doesn't do that, so there's no point of having a receptor in the muscle. How about our adipose cells? So how about fatty acid breakdown? Here, right, we do have your glucagon receptors, because now we actually want to use the same system here to actually increase cyclic AMP and generate the lipases so you can break down your fatty acids, because you need to break them down so you can run your TCA cycle and you can run your ETC and make ATP. So here again, it makes sense to have a glucagon receptor. How about insulin then? Right? Insulin actually works differently, right? It is basically like the worst enemy of glucagon. It is our feast hormone. It's a high glucose sensor. So it does the opposite of glucagon. It has actually its own receptor. It doesn't go through the same type of receptor. We have it in all organs actually, right? This has nothing to do with releasing glucose. We are actually wanting to trigger the opposite. So insulin receptors are present in liver, muscle, and the adipose cells. I don't ask you to really look at the mechanism itself. What I want you to note is that insulin not only works through different receptors, it actually is using a different kinase cascade. It's not using PKA, it's using PKB, protein kinase B. That protein kinase works with a similar mechanism, but it's a different cascade pathway. It increases glycogen synthesis, right? We have high glucose sensed by insulin. We want to get rid of the free glucose in the bloodstream. One way to do this is to produce glycogen. At the same time, you're reducing GNG and any other reaction that would release free glucose. What it actually does is that this phosphorylation cascade uses actually some of the same enzyme mechanisms, and what it does is it essentially undoes everything that glucagon is doing. All right, here just summarized what our hormones do, sort of in one table. Right, we have our adrenaline, which is our fight or flight hormone. The target cells, among others, but primarily is the liver, the muscle, and the adipose. It will increase abundance and activity of adenylate cyclase. That in turn will increase the abundance of cyclic AMP. And that increase in CMP abundance will increase and will trigger our PKA cascade. The exact same cascade is occurring in response to glucagon as a hormone. This is our low blood glucose sensor, right? Glucose gone, but it only occurs in the liver and the adipose, not in the muscle, because the muscle isn't releasing free glucose. And then opposite to that, our insulin, our feast hormone, our high blood glucose sensing hormone is again occurring in all of these three cell types, but it works via a different protein kinase cascade via PKB, protein kinase B, and it opposes all the mechanisms that glucagon is triggering. Okay. So what it actually does, we won't cover it, but what it does is it's actually activating a phosphatase that is removing all the phosphoryl groups, so it's inactivating all the products that PKA has activated.